Hello dear learners, in this video we are going to talk about what are restriction enzymes and what are their various types and their role in the genetic engineering. We will also learn some of the examples of these restriction enzymes. So first of all we should know that the restriction enzymes are the DNA cutting enzymes, and these DNA cutting enzymes are produced naturally by the bacteria. Now, why are they produced by bacteria? Because bacteria use these restriction enzymes as their defense mechanism to fight against the bacteriophage. Now, we know bacteriophage are the virus which infect the bacteria by inserting their own DNA into the bacteria. So now, to combat this, the bacteria produces the restriction enzyme innately, and these restriction enzymes are the DNA cutting enzymes and will help to cut down the DNA of the bacteriophage. So restriction enzymes are originally produced by the bacteria for the defense. But now we are using this in our biotechnology, in our gene cloning, recombination DNA technology for extracting our own purpose. So these enzymes recognize, first important thing is that they recognize very specific nucleotide sequence in the DNA. So there is a very specific sequence to each restriction enzyme, and they are cleaving the phosphodiester bonds only. So that means that whatever the two nucleotides are attached, there is a phosphodiester bond between them. So this is cleaved by the restriction enzymes. Now sometimes they are also called as restriction endonuclease because in the entire length of the DNA they are cutting in between. They do not cut at the end, or they do not remove the nucleotides at the end. The endonucleases are the ones which are cutting in between the length of the DNA. Now the restriction enzymes discovery made the recombination DNA technology very possible and easy. Now there are many kinds of restriction enzymes, but out of all those, type 2 restriction enzymes are most important ones. So basically we are going to talk about type 1, type 2, type 2s, type 3 and type 4. Now most of the restriction enzymes require ATP for their functioning, and physically and in our experiments we cannot always provide ATP for the functioning of the restriction enzymes. However, these two are the enzymes which generally do not require ATP for their functioning, and that's why we say that they can be used in the recombinant DNA technology experiments. Secondly, the other restriction enzymes cut the DNA very vaguely. That means that whatever their specific point of cleavage is, they will not cut at that point, and they get cut randomly within the specific range of the nucleotides. So again, this is not very fruitful. If we want to extract out a very specific sequence, then we should know that on this specific nucleotide only, if the restriction enzyme will act, then only we can obtain a specific sequence which we want. And type 2 and type 2s have that specificity which makes them very important. So as I already told you, these enzymes are those which recognize specific DNA sequence. Along with that, these DNA sequences which they are identifying are also referred to as recognition sequence. And within the recognition sequence, they identify a particular point where they have to act. And this particular point is called as recognition site. Now the recognition sequences which they identify, mostly, they are not always, but mostly they are palindromic. Now suppose there is a double stranded DNA, one is running from 5' to 3', the other is running from 3' to 5'. So what do we say is that if here it is cutting between A and G, so here also it has to cut somehow between A and G. If here it is cutting between C and T, so somehow here also it has to cut between C and T. Now if you are reading from 5' to 3', so first A will come, then G will come, then C will come, and then T will come. And from here also if you are reading from 5' to 3', first A, then G, then C, and then T. So this is a palindrome, and that is what they recognize. This palindromic sequence, mind you, is generally of 8 to 9 base pairs which they recognize. This is a specific range which most of the restriction enzymes follow. So there is an example of palindromic sequence which is recognized by EcoR1. So you can understand it for your simplicity, or you can learn this example. Now there are more than 3700 type 2 restriction enzymes which have been discovered, and they are acting on more than 250 different recognition sequences, and they are also of varying length. Now we also need to know the first of such restriction enzyme which was discovered and was characterized was EcoR1. Now we should know that how this EcoR1 is named. So this E and Co, this E stands for the genus name, that is Escherichia, from where it is isolated. So we know that E. coli is a bacteria, and from this E. coli this restriction enzyme has been isolated. So E stands for the genus, Co stands for its species, that is coli. Then R stands for its strain. It can be a rough strain, it can be a smooth strain, or any other thing. 1 stands for the number of discovery. For example, at the very first instance, if it was discovered, so it was termed as 1. If it was discovered later, it might have been named as 4, 5, and whatsoever. So this is a genus 1, this is species, strain, and the order of discovery. Now let us try to learn the types of restriction enzymes. As I told you, there are 4 to 5 kinds of restriction enzymes. So we need to know about them in detail. Let's first talk about the type 1 restriction enzymes. Now, as you study, you'll come to know that there is another function of the restriction enzymes also. Apart from just cutting at the recognition sites, the restriction enzymes can also do the methylation activity. And out of the 5 restriction enzymes which we are studying, there are many of them which follow both the things simultaneously. For example, type 1 restriction enzyme will have the methylation activity also, and will have the restriction activity also within the same enzyme. Now if I talk about the position where the type 1 is going to cut, so it generally cuts far away from the recognition site. Suppose this was a particular sequence, and if here was the recognition site, so it will not cut here, but it can cut in any range here, and roughly it is within the 1000 base pair range. So it is a very vague cutting, and we cannot be completely trusting upon that what kind of sequence will be produced, because every time it can vary. Third thing, that it requires ATP for its functioning, it requires magnesium ions, and it requires S-adenosine methylase. So this is also one of the things which the type 1 restriction enzyme requires. You can also just confirm the full form of this SAM, but it's roughly the same. Nevertheless, the three of the things are required by type 1 restriction enzyme to function. One of the examples of type 1 restriction enzyme is EcoK, but it is not much used, but you can still learn the example. The second type of restriction enzyme is the one which is unifunctional in nature. It means that either it can have restriction activity, or it can have the methylation activity. It generally recognizes a palindromic sequence, and then only it will act upon. It cuts at the very defined position, and always it will do the same thing. So it can be trusted upon. It do not require any ATP, it do not require any SAM, it only requires magnesium for the functioning. The examples can be EcoR1, one of the earliest discovered restriction enzymes which is still in use. The example can be BAMH1. The example can be HIND3. So all these are important ones. HIND3, Haemophilus influenzae. And these are very important and useful in genetic engineering. Also, in the continuation, we should first talk about type 2S restriction enzyme. So it is also a subgroup of the type 2 restriction enzymes. Now what does it do is that although it does not cut at the specific recognition site, but it will cut a bit far away. But that far away position is always fixed. Suppose if it is cutting 6 base pairs away, so whenever you are using this on that particular DNA, it will always cut 6 base pairs away only from its recognition site. So in that way also it can be trusted. Secondly, it only requires magnesium for its functioning. ATP is not required, SAM is not required, so still it can be relied upon. The example is FOC1, and we will even see in the zinc finger nucleases and even in the talons that Fork1 is very important restriction enzyme used there. It is used in advanced cloning methods. Now, what is the meaning of cloning? Cloning means when you are taking a plasmid, you are cutting it with one restriction enzyme, and then you are taking a foreign DNA and you are cutting with the same restriction enzyme, and then you are putting this particular part here. And now, if it starts replicating so that the foreign DNA copy increases, so this replicating part to increase the number is what we call as cloning. So for cloning, we need to cut the particular sites, and for this fork1 enzyme is very popularly used. The next important enzyme is type 3 restriction enzyme. Now it has the combined function of restriction and methylation. Since this function is not required, we cannot trust it. Secondly, it cuts a short distance away from the site, and that is also not guaranteed where it will cut. Thirdly, it requires ATP, so here only it is cut. SAM is although not required, but ATP is still required. So we cannot be relying upon this. And then the example includes EcoP15. The next is type 4 restriction enzyme. And the main function is not the cutting one, but it recognizes any methylated DNA. And it can do cleavage, but the cleavage is very variable. It does not require SAM. It does not require magnesium, but it requires ATP. And here only our criteria doesn't fulfill. The example is MCRBC, which can be one of the type 4 restriction endonuclease. So what I want to say in conclusion is that out of these 5 restriction endonucleases which we have studied, type 2 and type 2S are the most important ones since they do not require any ATP, since they do not have the methylation activity, and also because they cut at very specific positions. Hence, they are very important in the genetic engineering and cloning experiments. So, as I told you, and here it is written in continuation, the cleavage in type 2 restriction enzyme occurs very close to the recognition sequence, and generally the recognition sequence is of 4 to 8 base pairs. Always remember, if you are using, suppose if I say BAM-H1, now if this BAM-H1 is used on a specific DNA any time of at any point of time, it will always generate the same identical fragment. No variation would be there because its restriction site is very fixed. Now these type 2 and type 2s restriction enzymes can produce two kinds of fragments after cutting. One of them can be sticky fragments and one of them can be blunt with blunt ends. So if the sticky ends are produced, so let's take an example. If this is our particular DNA, now here it has been cut and here it has been cut. Now it is cut in a way that such kind of DNA is produced. Now here you have a sticky end and here also you have a sticky end. But if it is cut in a way that, let's say if it is cut like this way, in that condition this kind of DNA will be produced with no sticky ends here. So either the sticky end can be produced or the blunt end could also be produced. So there are generally 5' phosphate overhangs when cut by eco R1. Means after 5' also are certain sequences which are the overhangs left over when cut by eco R1. Now these sticky ends are very important because they can easily facilitate the joining of that particular DNA into the vector. But if you take a restriction enzyme which is producing blunt end, for example HIND2, for example HAE1, so these will produce blunt end, although they will not help us to easily ligate that DNA, but still they are used somehow. Now there are some type 2s restriction enzymes as I told you, and the important example being fork1. This fork1 recognizes the sequence GGATG and it cuts 9 nucleotides downstream. Now see, let's say if we are taking a DNA strand, here 5' to 3' and here 5' to 3'. So from this, suppose if this is the recognition site, so from here 9 nucleotides downstream, Here it will actually cut on one strand, and on the other strand also it will cut at 13 nucleotide downstream. Suppose here was its recognition site, so it will cut it 13 nucleotides later. And because this thing is fixed, so we can trust upon this that it will always cut at one strand 9 base pairs, and at the other strand it will cut at 13 base pairs distance. And they are widely used in genome editing systems, for example in zinc finger nucleases and in talons. So we will also study about them. Okay, I already told this part that how bacteria use these restriction enzymes for their protection, and now there are some of the special categories of restriction enzymes. The categories are isoschizomers, neoschizomers, isochordamers. So if we take two restriction enzymes derived from two bacteria, since it is bacteria 1, let's say, and it is bacteria 2, maybe from different species, we are taking restriction enzyme 1, restriction enzyme 2. If both of the restriction enzymes, when acted upon a particular DNA strand, recognize very similar strand or recognize the exact same strand and cut at very same sequence only, then the two restriction enzymes are isoschizomers of each other. What I want to say is that although you are isolating the restriction enzymes from two different bacteria belonging to two different species, but if the two of them are acting very similar to each other, they are cutting at a very same position, then we say that they are the isoschizomers. And one of the important example is of SVH1 and BBU1. Similarly, it is XHO1 and PAER71. Then there are neoschizomers. Neoschizomers, although they recognize the same sequence, they come to the same sequence, but they do not cut at the specific position. For example, this is our DNA. Both of them have identified that they have to cut here. But let's say one of them is cutting here, another one is cutting here. So although they are producing different tails, but they are recognizing a same region, and they are categorized as neoschizomers. The example is SMA1 and XMA1. They recognize this sequence. Now ideally it is a palindromic sequence only, because it should be designated this way. So they are recognizing this particular site, but maybe one is cutting here while the another one is cutting here. Depends upon. Third is isochordomers. Now they will recognize very different sites. No, not at all similar sites. But the tail which they are generating will be similar. Suppose one of them is generating a trithymidine tail. The another one is also generating trithymidine tail only. Although here it can be anything, and here it can be anything, but still they are generating a same kind of tail, then they are isochordomers. From chordo you can understand and you can learn it with the help of this mnemonic, that chordo is something related to tail. The example is BAMH1 and also SAU3A1. Roles are very clear. One of them is to form the recombinant DNA. The another one is for the gene cloning. As I already told you, type 2S restriction enzymes mainly used for this only. Then there are restriction mapping also. You can identify the restriction sites in a particular genome by single digestion and performing double digestion. Suppose you are taking two restriction enzymes R1 and R2. So one time you take only this and you let it act upon this. At the second instance you only take R2 and let it act upon this. And at the another instance you take both of them and let them act upon this particular DNA. And then you, whatever fragments are generated, you do the gel electrophoresis of that, and you will easily come to know that how are they acting and what are the exact sites, what are the exact locations of those restriction sites. You can even do the analysis of the DNA fragments, genomic analysis and all that. DNA ligase also here adds on, because once you are cutting and suppose it is producing blunt ends, now blunt ends are not self sticky, so you will require the DNA ligase to act upon. and one of the important examples of DNA ligase is T4 DNA ligase. So that's all for the restriction enzyme.